Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about protein folding and molecular chaperone. This molecular chaperone are also called as heat shock protein. In short form we can say HSP. We will explain it that why we call that heat shock protein. Now in the previous series of video lecture, if you guys remember, we talked about the protein synthesis in prokaryote as well as in eukaryote. Or we can say we talked the translation in prokaryote as well as in eukaryote, but we didn't talk that once the protein are synthesized, means once the polypeptide chain are synthesized, then what happened with this polypeptide chain we didn't talk about. You know what happened during translation, termination of translation, once the protein are synthesized, so we get a linear structure of the protein. You know, sometimes this protein are also called as polypeptide chain. Why we call that polypeptide chain? We call that polypeptide chain because in this polypeptide chain there are in the protein there are a lot of amino acids which are connected with each other through a polypeptide chain or polypeptide bond. So when amino acids are connected with each other through a polypeptide bond, then we call it polypeptide bond because there are a lot of peptide bond, and due to this way we are also called it polypeptide chain. Okay, so during translation, after translation, what happened? The protein are synthesized, so we get a linear structure of the protein. This linear structure of the protein are non-functional, and this linear structure of the protein doesn't have any function. It means, and one thing more, this linear structure of the protein we also call that nascent protein. And it means that nascent protein means newly formed protein which don't have any function. So what happened? This protein should be converted into a functional. So the protein which is synthesized are also in an unfolded form, means it is not folded. So this protein will be converted into a folded form. And the second thing is that this protein are also converted into a secondary or tertiary structure. Once the protein are converted in the non-linear form, so then this protein will be functionable, or it will be perform their function. And one thing more, this protein are called as a native protein inside the cytoplasm. So how it is converted? This is the question arises in our mind, that how the linear structure of the protein are converted into a non-linear structure of the protein or into a secondary tertiary structure of the protein. So it will be converted with the help of molecular chaperone, or we call that a helper protein. So how the unfolded form of the protein will be converted into the folded? It will be possible by a molecular chaperone, or which act as a helper protein. And one thing more, that why we call that heat shock protein? We called the molecular chaperone, we called it heat shock protein because it was believed that each protein was working at a very high temperature and pressure, and once it exposed to a toxic and chemical or radiations, due to this way the chaperones, 20 different chaperones are activated. Due to this way we call that heat shock protein. And whenever there is no stress or other condition or heat other condition, then we can say still the chaperone are activated, but very less amount of chaperone are activated. You know, at a temperature of 30 to 42 centigrade inside the E. coli, so 20 chaperones are activated in just 5 minutes. And if we talked about the heat shock protein in amount, so almost there are a lot of, or a large amount or large quantity of heat shock protein in case of hypothermal bacteria, like pyrodactum occultum bacteria. Okay, like pyrodactum occultum bacteria. So such bacteria can grow at a very high temperature, like 110 centigrade. So in these kinds of bacteria there are a lot of chaperones. Now this is general introduction of the molecular chaperone and protein folding. So remember guys, when the protein is in a newly formed protein, then we call it nascent protein, but when it's converted into a folded form, then we call it native protein. And you know what happened inside the cell cytoplasm? There are a lot of proteins are present. Unfolded form of the protein means the primary structure, means linear form of the protein, means unfunctional protein are present. So The chaperone will only bend with these kinds of protein. It will not bend with the folded form of the protein. It will not bend with the functional protein. It will not bend with the secondary, tertiary or quaternary structure of the protein. It will not bend with the non-linear structure of the protein. It will bend with the unfolded protein. And once it bend with it, so then it will convert it into the functional protein. Okay. And one thing more. This chaperone will also bind with the incorrect folded form of the protein and make it in a correct folded form. So this is all about the molecular chaperone and protein folding. This is just the idea, or we can say this is the overview. This is just the introduction. Okay, overview. So this is the overview, and this is the big picture of the protein folding and molecular chaperone. So if we talked about it with the help of diagrams, so what actually we have, this is, let's suppose, the messenger RNA, as you can see, and this is the ribosomes. This is the large subunit, this is the small subunit of the ribosome, and this is the protein are synthesized, as you can see. So once the protein are synthesized, so it will be in a linear form, as you can see. So this is the linear form of the protein. Okay, so this is actually the linear form of the protein, as you can see. Now these circle, as you can see, these are actually the amino acid, and all of you better know that inside the protein we have, at one end of the protein we have amino group, which is the N terminus, and at the other end of the protein we have the carboxylic group, which is the C terminus. Okay, so each protein are made up of with amino acid, and each amino acid are connected with each other through a peptide bond. That's why we also called it polypeptide chain. Now this is the linear structure of the protein, and this linear structure of the protein don't have any function, and this linear structure of protein are actually in unfolded form, as you can see. So what happened, it will be converted into a folded form. But before that, what happened, inside each protein there are two sides of the protein, as you can see. So let me label. So now these green color, as you can see, these green dots are actually the polar side. These are the polar side. So these are the polar side of the protein. And these blue color, as you can see, these are the non-polar side. So it means that protein have two sides. One is the hydrophilic side. It means that one is the hydrophilic side. It means that it will be, make a bond with water molecule. It also means that it will easily soluble in the water. And this hydrophilic side are also called as polar side, while the other side is known as the non-polar side, which is the hydrophobic side. It means that it is not solved inside the water, or what we can say, it can make a bond with water. So during folding of this protein, the hydrophobic side will get internalized, while the hydrophilic side will be, remain outside, due to which it will be easily soluble. So what happened, we have a molecular chaperone which are involved in this case protein. So heat shock protein are involved in this case, and they will convert this linear chain of the amino acid or linear chain of the polypeptide into a folded form, or into a secondary structure, or into a non-linear form, as you can see. So what we have, so this is the folded form of the protein. This molecular chaperone or heat shock protein will convert the primary structure of the protein into a secondary structure, and this secondary structure of the protein, sometimes we call it alpha helix, or sometimes we call it beta plated sheet. So let me write. So it will be called alpha helix or beta sheet. So this is how the structure of the protein we have. So as we can see, these are the polar side, which is outside present, and these are the non-polar side, as you can see, which is present inside. So this is the folded form of the protein. Okay, now this protein are functionable, and this protein is now can perform their function. Okay, so now let's talks about the molecular chaperone or heat shock protein. Now, if we talked about the heat shock protein or molecular chaperones, so remember guys, we have a lot of heat shock proteins. We have 20 to 70 or heat shock protein we have inside a cell. So almost we can convert the molecular chaperone or heat shock protein into 5 different classes or 5 types, what we can say, whatever we can say. So each heat shock protein or molecular chaperone have the same function, but their naming are different. Okay, so let me write. We have chaperone 1, chaperone 2, chaperone 3, chaperone 4, and chaperone 5. Okay, in prokaryotic cell, what we call that chaperone 1, what we call chaperone 1, and in eukaryotic cell, what we call chaperone 1. So remember, in prokaryotic cell we have DNA K, which is mentioned in chaperone 1, and this DNA K inside the prokaryote we call that DNA K, while inside the eukaryote we call that heat shock protein 70. And we have also DNA J, so we call that heat shock protein. And we have also grip E grows. Okay, so this grip E, this grip E is called as heat shock 60, and this groove, it is also called heat shock protein 60. And we have also another class of the chaperone, 5th class of the chaperone, we call it groove ES, so we call it heat shock protein 10. So let me a little bit explain that what are actually these of the things. These are actually the types of the molecular chaperones. Now we have chaperone 1. In chaperone 1 we have DNA K. Inside the prokaryote we call that DNA K, and inside the eukaryote we call chaperone 1, we call that heat shock protein 70. Now we have chaperone 2. So chaperone 2 in case of prokaryote we call that DNA J, while in case of eukaryote we call that heat shock protein 70. Now then we have chaperone 3. In case of prokaryote we call that grip E, and in eukaryote we call it heat shock protein 60. Now then we have chaperone 4. In case of prokaryote we call that grow EL, while in case of eukaryote we call that heat shock protein 60. And 5th we have chaperone 5, we call that grows, means grow ES, and in eukaryote we call that heat shock protein 10. So this is a little bit types. Now then we have some other types of the protein, we call that on the basis of their activity we have some other type of protein like Holdases, foldases, and disagregases. So these are some other types of the protein. As the name indicate, Holdases are those kinds of the molecular chaperone which are help the protein to hold or to prevent it from the degradations. So there are a lot of chaperones, molecular chaperones, which are responsible to prevent the protein from the degradation. So in Holdases, the example are DNA J. In prokaryotes we have DNA J, while in eukaryotes we have HSP40. Okay, then we have foldases. Foldases, as the name indicate, these are those molecular chaperone which are responsible to make the protein into a folded form. And in this case we have the example, grow EL or HSP60. And then we have disagregases. So disagregases are those molecular chaperones which prevent the protein from the aggregation. Now once the protein, remember, once the protein are in an aggregated form, so it will be converted into a prion. Prions are different kinds of disease then it will cause. Okay, and in these, if we talked about disaggregates molecular chaperones, so the example are HSP10 in Eukaryotes or GROES, GROES in prokaryotes. So these are some other types. Then we have some other chaperone, we call that Lectin chaperone. So let me write. So there are two types of the Lectin chaperone. One we call that Calnexin. So let me write. While the other we call that Calreticulin. Calreticulin. Okay, these two are actually involved in calcium or regulate in calcium IP3 pathway. Both these two lectin chaperones are involved to make the MHC1 class protein. MHC1 class protein. Now what happened if we talked about the protein folding phenomena? So we have almost three pathways of the protein folding. So we have three pathways of the protein folding. One we called it on pathway folding. Other we call that off protein folding, off pathway folding. And third we call that proteosome system. So this is the pathway of the protein folding. You know what happened if the linear structure of the protein is converted into a non-linear structure, or if the unfolded form of the protein is converted into a folded form, which is functional in the future, then what happen, this protein will perform their function. Sometimes this protein will be not converted into a folded form. So after that what happen, if the protein are not converted into a folded form, it will create a lot of problems, or sometimes it will create prions like disease. So what happen after that, this protein which is not converted into a folded form, it will be shifted to the proteosome enzyme. Proteosome is a kind of degradative enzyme, so it will degrade this protein. And once the protein are degraded, so amino acid are formed. So each amino acid are again connected to each other. So then amino acid, so sequentially the amino acid are connected with each other again. And after second time, this protein is now in second time this protein will be converted into a folded form. Okay, so this is all about the molecular chaperone and protein folding. Okay, and these are also the function of molecular chaperone and protein folding as well. One thing that I forget here, we have also another chaperones, we call that Sec-B. This Sec-B is also important in Sec-dependent pathway. In this case what we have the unfolded form of the protein, as you can see. So the Sec protein will bind here, as you can see, and it will just export this protein from the membrane. So if this is the cell membrane, as you can see, so it will be, this is the protein which is formed inside the cell, and it will be exported this protein outside the cell membrane, as you can see. So this is the protein, and how it will be eliminated from the cell through this Sec-B. And this system is called a Sec dependent system. So this is all about the protein folding and molecular chaperone. Inshallah, in the next video lecture, I'm going to talk about the mechanism of these molecular chaperone during protein folding, that how it will perform their function. And we are going to talk about each mechanisms in case of prokaryote and in case of eukaryote. So the mechanism are simple in both, means are same in both, but the naming are just different. We will be talking all these things in the next video lecture. So stay tuned and keep watching. Thank you so much for watching this video.